Hello there, I am doing my video about the basics of inheritance. So, let me first explain the basics of inheritance by discussing what dominant and recessive alleles are. Alleles are different forms of a gene. Dominant alleles are more distinctive than recessive alleles. Therefore, the dominant alleles for a trait disguise the recessive alleles. Gregor Mendel's experiment of pea plants revealed that certain traits are either recessive or dominant, and their offspring will inherit one or a mixture of these traits. When two alleles are joined together through mating, the dominance versus recessiveness for a trait can either be dominant and recessive, recessive and dominant, or dominant and dominant. These factors determine which traits their offspring will inherit. When an allele is dominant, whatever trait it is connected to will be expressed. When one allele is recessive, the likeliness of the characteristic being expressed is less than if it were dominant, because the recessive traits are only expressed when both alleles are recessive. Genetic makeup and genetic expression are related to dominant and recessive alleles. Phenotype are the obvious characteristics that someone has, like hair color and texture, skin color, height, etc. It is determined in combination of environment and the particular set of genes the individual inherited from the parents, which relates back to recessive and dominant genes. Genotype is the genetic material that an individual carries and inherited from their parents. These genes come in pairs. One is inherited from the mother and the other from the father, dominant and recessive for a trait. So now let's discuss what genetic disorders are, what causes them, and how some offspring in a family inherit a genetic disorder and others in that same family do not. A genetic disorder is a disease caused by a change in the DNA strand, which interrupts its normal sequence. This abnormal sequence is due to a mutation which expresses itself by either monogenic disorders, which are a mutation within only one gene, multifactorial inheritance disorder, which is a mutation involving multiple genes or multiple factors. It can be a combination of gene mutations and environmental factors, and it also can be damage to chromosomes, changes in the number or structure of the entire chromosome, which are what carry genes. Changes to a chromosome during meiosis, such as an environmental factor, as carriers from the carrier mother or father being exposed to something that caused the mutation in the chromosome. Gene mutations occur during the chromosome division process called meiosis. There are two processes of division, meiosis and mitosis. Meiosis is the process of cell separation where four genetically different sex cells called gametes are produced. During genetic recombination, four genetically different sex cells are produced. Mitosis is the process of cell division where two genetically different body cells or two genetically identical body cells are produced. Within these cells of the chromosome is its genetic material or code. This process occurs in order to replace dead, damaged, or short-lived cells. What is the difference between gene mutation and chromosomal mutations? The only way brand new genetic material is created is through gene mutation. There are mutations that occur within a gene, Mutations that occur within a chromosome. Differences between gene mutation and chromosomal mutations are that gene mutation occurs as a change in the nucleotide sequence, such as sickle cell anemia. These mutations change the shape of blood cells, which prevents normal levels of oxygen to reach the cell. Chromosomal mutations are a change in the structure or number of chromosomes, which is what causes disorders such as Down syndrome. During crossover in meiosis, a chromosome is copied and duplicated, which results in an extra chromosome, 21, and no matching pair. Sometimes a chromosome can get lost, which leaves the organism or individual with one fewer chromosome, and no matching pair, which is what happens in Turner syndrome. Gene mutations can come in the forms of point mutations, which is a change in a single nucleotide substitution which is a substitution in the DNA strand. Base pair insertion or deletion are where base pairs are either inserted, which is added, or deleted from the original DNA sequence. When stop codons are introduced into the sequence either too early or too late. That's what causes a gene mutation. Chromosomal mutations are changes to structure, translocations, exchange of chromosome sequences between non homogeneous chromosomes. Duplication is the production of extra copies of genes. Inversions are broken sequences of chromosomes are inverted or inserted in the same position. Okay, now let's talk about types of point mutations. Point mutations can come in silent mutations, which is that even though a change in the DNA sequence has occurred, it does not affect the protein and the production of the code for the amino acid. I don't know how to say this word. Missense mutation. The nucleotide sequence is altered and a different amino acid is produced, therefore the protein is affected. Nonsense mutation alters the nucleotide sequence and a stop codon replaces the amino acid. When this occurs, it stops the protein production. If the protein production is ended too soon, it will cut off the amino acid sequence, which results in a non-functional protein. And here is a diagram of a point mutation. Okay? And my references. Thank you.